So I think you can start.
Thank you, Kim. Thank you, everyone, for coming. So this is the fourth and last lecture in this series on quantum biology. Sadly, I'll only cover about half of my slides, maybe less. But I think we covered a lot of territory. It's not about how many slides, but what kind of concepts and how deep we can go into it. So today, I will try to cover or discuss two aspects related to quantum biology. The first one is quantum metabolism. It's very important because, as I emphasized a few times earlier, metabolism is one of the distinguishing features of living systems. And if this is quantized, then of course, the foundation of life is quantized. The second is even maybe more important, is the concept of intelligence, cognition, consciousness, information processing, these kinds of things which have to do with intelligent behavior of living systems. And so I'll start with this, and then we'll move on to the other topics. And that kind of is the basis and has some central aspects. A single cell is not just a bag of proteins in a fluid, but it is actually an intelligent system that responds to stimuli and sometimes will intelligently seek nutrients and avoid potential problems, obstacles, toxins, life-endangering situations, and of course will seek mates if it's a cell that reproduces by mating. So where is this coming from? Where is that processing of some sort of inputs and generation of outputs taking place? An interesting person, a theoretical physicist by training and turned cell biologist, Günther Albrecht Bühler at the Northwestern University of Chicago, spent about 2 decades trying to understand and answer that question. And he claimed that the cell can be thought of as an intelligent organism already, in its all entirety, with the powerhouse mitochondria producing the necessary energy, the library DNA of all the instructions to make proteins, and also the muscles and the bones, cytoskeleton, microtubules, actin filaments. And then most importantly, of course, the membrane as well, a bit like skin for protection, ion channels for selective passage of nutrients and ions, etc. But most importantly, in his opinion, the centriole is the command center, if you will. It's an eye and the brain of the cell, according to Günther Albrecht Bühler. And so I'll spend a few minutes talking about how that is envisaged. And he did some very clever experiments, which were intended to illustrate that cells interact with other cells, perceive other cells, communicate with other cells, not by physical or chemical means necessarily, but by electromagnetic means, by sending electromagnetic signals, in other words, photons. So we're back to early quantum mechanics. And that's why I put this topic inside the series, because it has to do with potential quantum communication between cells. And he constructed his laboratory setups in such a way that there was no way that the cells could communicate by some hormones or enzymes or any chemical means, or even physical contact, because they were separated, and they were still demonstrated to perceive others, move towards other cells. And then he actually went one step further and tried to identify the type of electromagnetic frequency which would involve, or wavelength, which would involve this signaling, and kind of fool the cells by shining artificially created light, which was in the near infrared and visible range, and that was reflected from a bead, and then he noticed this specific reaction of the cells to light, and that diagram on the right shows you the peak sensitivity at around 900, 850, 900 nanometers, which is actually not visible. It's in the near infrared, so it's not visible to us, but possibly visible to cells, or in fact received by cells, transformed in some way, and processed. So the question, several questions arose. The first one is, what is the structure in the cell that may detect this kind of electromagnetic signal, first of all? And the second question is, on the other side of this equation, what is the structure in the cell that can emit electromagnetic waves? In other words, emission and transmission and absorption. These are the three issues. The transmission is easier to address because these are micrometer distances. Electromagnetic waves will not lose a lot of energy. Longer, perhaps, distances would be an impediment. So he pointed out to centrioles. The centrioles have a very intricate structure. These are, I should say, centrosome. Centrosome is two centrioles. If anyone here is a cell biologist, you will know already. So two centrioles oriented perpendicular to each other, typically in cells, form a centrosome, and that centrosome is surrounded by pericentriolar material with some proteins, and it's an organelle which is actually also a microtubule organizing center. In other words, microtubules grow radially from centrosomes, and in dividing cells, this is how cell division process originates. The first step in cell division is centrosome duplication. First, centrioles segregating or separating from each other, and then growing partners at right angles, and then moving to opposite positions, polar positions inside the cell, and then growing microtubules from them radially in sort of astral orientations. And then in the meantime, you have the chromosome, the nuclear envelope, dissolving chromosomes formed in the middle plate, mitotic plate, and microtubules connecting. So the details are very well known to all biologists, but the question, of course, is, what is this first trigger for cell division? Where is this coming from? And why is this, and why and how this centriolar separation and duplication occurs? It's a kind of mysterious mechanism. In fact, there's a book called The Centriole, the Key Enigma in Cell Biology. I think I'm just paraphrasing the title, something like that. It is an enigma still. And so according to Günther Albert Bühler, maybe I'll stay here. The centriole is an important nanotechnology device, actually, because it is allowing the cell to not only absorb this light and trigger subsequent reactions. We think it's electron transitions which lead to mechanical transformations and so on. But very importantly, because of their perpendicular orientation and actually the architecture of the centrioles, composed of 9 triplets of microtubules, as you can see in this diagram, which are somewhat mechanically movable, and their positions can be adjusted. The centriole can detect light to within something like 5 degrees in both directions, azimuthal and out of plane. So it allows, it's almost like a detection mechanism for the source of light. And therefore, if the source of light is not the cell, that cell that detects it will be able to orient itself towards the source and move, trigger the mechanism of mechanical transduction and movement, cell motility, basically, through microtubules, actin filaments, and so on, motor proteins. So that's kind of very, very neat. And I have no time to go into deeper details, but there's a very nice website that this professor created with probably more than 100 pages of illustrations from his work. It is actually called Cell Intelligence. Cell Intelligence, or if you just Google it, I think it'll take you there, and you'll see a great description of his experiments and the conclusions. The remaining question still is, where is light coming from in a living cell? And so I did some work in the past, and with my colleagues, we hypothesized and demonstrated from physical principles that the functioning of mitochondria with the proton transfer across the mitochondrial membrane and the cytochrome enzyme is coupled to the emission of a single photon. So one transfer of proton causes an emission of a single photon. And actually, we demonstrated that this would be in the range that roughly corresponds to what Günther Albrecht-Bühler found experimentally. There are some other possibilities like reactive oxygen species recombination, but that would be a bit too, I would say, left to chance. If this is just random processes of recombination of reactive oxygen and nitrogen species, then it's all kind of random. Here we have something which is linked to the rate of metabolic reactions, which we'll come back to, and therefore a living cell would be sending signals, I'm alive, and sending photons in the direction all around, but if there's another cell, you could draw a straight line between the two and they will know which way to go. So let me just move on. So this is, there are some consequences of this kind of thing beyond, of course, the potential answer to the question, where does intelligent behavior consciousness begin? This is like a proto-conscious system, which uses electromagnetic waves for communication and information gathering and processing. And there are some very interesting experiments at higher levels, cell cultures, where, for example, it's well known in cancer cell biology that there is something called the bystander effect, where in a culture of cells, millions of cells, hundreds of thousands, some will be hit by radiation, and they will respond by triggering repair processes. And it has been demonstrated that even those cells which were not hit, they do the same. They kind of get the information from the surroundings that something is happening, something untoward. We're being bombarded. Let's hunker down, or mobilize for damage repair. So that's called the bystander effect. And of course, the question is, how do they do it? If they could be far away from one another, and that would be one possible answer, that signals in the form of electromagnetic, maybe some kind of SOS code. We don't know yet. Maybe sent out and received and acted upon. So that actually relates not just to radiation damage, but also stress. Any kind of stress applied to some cells will be perceived by others in the vicinity. I have a colleague of mine at the University of Central Florida in Orlando who did experiments on providing nutrients to cells, and they were separated physically, and cells which received nutrients upregulated their metabolism, and cells which were in vicinity didn't get the nutrients, they also did the same thing. So there's some kind of synchronization. We don't know exactly what, but if we put this together with Günther Albrecht-Bühler's work, that would make sense, that they communicate electromagnetically. There is a very brilliant radiation cancer cell biologist, Carmen Mothersill. Actually, I think she's a holder of Canada research chair at McMaster, and she has observed and published these kinds of bystander effects going beyond cells, actually to the entire organisms, and published some amazing results, including the following. So she did experiments with radiation exposure by two groups of fish, which were swimming in the same tank, but they were separated physically. One was irradiated and the other not. And their response in terms of heat shock protein activation and so on was also communicated. So both groups acted the same way, even though the second was not exposed. So that is kind of interesting. And there was even a more fantastical experiment which was published and left without an answer, but she measured these responses even prior to administration of radiation, and the fish reacted before irradiation. In other words, there was some sort of signal traveling in time, forward in time. So how do you explain this? Well, I think using quantum mechanical processes might shed some light. And in fact, Professor Mothersill implicated quantum interactions. The spooky interaction at distance, in this case, spatial-temporal interactions. In quantum mechanics, by the way, the solutions of quantum equations, be it Schrödinger or Dirac equations, are allowed both forward and backward in time, by the way. So there is no prohibition against moving back in time in quantum mechanics. If you go into statistical physics, that's where things get a bit dicey, because these are then contradicting the second law of thermodynamics, the increase of entropy with time. In quantum mechanics per se, you don't have that restriction. You have equations which are completely reversible in time. So I'll leave it at this. So if we can understand cells communicating electromagnetically, maybe via mitochondria emission and centriolar absorption, and then transduction into the cytoskeleton and causation of cell motility, that's fine. But how do you explain organisms as a whole responding to this? Then we're back to square one in terms of synchronization, maybe by some quantum condensation mechanisms, at least some degree. I'm not saying that every single protein in the human body is talking to the rest of the proteins, but I think in terms of some important degrees of freedom, some perhaps involved in information processing, we'll come to it, may be in this kind of quantum regime, or could be in a quantum regime. All right, these are the references, by the way, to these experiments on the bystander effect by the group of Professor Mothersill from McMaster. I don't know if we are sharing the, but it'll be on YouTube, so we can stop it at any time. Okay, but if anybody wants more information, please email me. Now, let me move on to the next topic. We are now moving to the macro scale, right? Macro scale from photosynthesis, micro scale, and so on. The next topic is quantum metabolism. So this is kind of an interesting and centuries-old, again, enigma or puzzle, a scientific puzzle, going back actually to some very famous people in science, Lavoisier and Laplace, late 18th century. Namely, what is metabolism? What does it involve? It's respiration, some form of linked to respiration, some form of combustion of energy given molecules and production of heat, maybe some products as well. But as we know now, it's mainly production of ATP and GTP, these kinds of energy carrying molecules in biochemistry. And then Rubner then later on Kleiber did some very careful studies of measuring the rate of metabolic rates. In other words, how much energy, how many calories a given organism utilizes as a function of size. And they looked at insects and vertebrates and mammals, elephants, humans of different sizes. And guess what? I mean, this was kind of surprising that the function of the metabolic rate does not scale with size linearly. You would expect that if, let's say, an elephant is, I'm guessing, 100 times heavier than a man, maybe more, it would require 100 times as much nutrients per day or per minute or hour. No, it doesn't. And that scales all the way down to even bacteria, actually, from bacteria all the way to the largest mammals. And what happened is that by and large, the simplest message, and there's a lot of messages here in this slide, but the first thing is you can measure the metabolic rate under different conditions, right? There is the basal metabolic rate, a field metabolic rate, which changes, it's higher, and the maximal metabolic rate. You can push somebody to the, let's say for humans, to maximum exertion rates and then measure what is the, but let's say we stay with the basal metabolic rate, BMR, and that rate scales typically with the 3/4 exponent. In other words, what it would be, if you increase the size 8 to 16 times, so let's say from a small child to an adult 16 times, from 5 kilograms to 80, the rate increases only 8 times, not 16, because it's 3/4. So 2 to the power 4 raised to 3/4 is 2 to the power 3. And that also applies across species. If you, let's say, compare the rate of a mouse to a rat, and the mouse will be 20 grams, a rat maybe 200 on average, something like this. So it's tenfold. It's not going to be 10 times higher rate. It'll be 10 to the power 3/4. Roughly speaking, it's 3/4 is 0.75, right? It's roughly 2/3, 0.66. And why am I invoking 2/3? Because 2/3 mass scales with volume, right? Because we are roughly our density, the density of the human body is equal to density of water, roughly, give or take. It's about 1,000 kilograms per meter cubed, right? The density of water. So if you scale the volume of the body linearly, so increase, let's say, the height by a factor of 2, then the volume increase by the factor of 2 to the power 3. So volume will increase 8 times, but surface area, body surface area, will roughly increase 4 times, right? 2 squared. So 2/3 is basically scaling with the change in the surface area. And 2/3 is not equivalent, but close to 3/4. So people thought that it has to do with the, well, there are a lot of hypotheses, maybe vasculature, because you have to deliver transport food to all the cells. So it has to do with the transport, and there is a famous fractal theory. I have no time to go into this fractal geometry aspect, but it's basically branching. Blood vessels branch out. There's like 12 different sequences of branching out from the aorta down to this sinusoid. So that's one explanation, but it doesn't explain things like single cell organisms, bacteria, or or single cells, or even small or organisms which do not have vasculature. So that fails on one account at least, but there's more. So that's a problem. There are some variations between plants and animals.
Question.
Yes.
I'm just curious, what does alpha represent?
Oh, yes, very good question. Right. So this is a very simple formula which tells you Y is the basal metabolic rate, W is the weight of the organism. Alpha is some coefficient of proportionality, which is very much species dependent, and this is where there is a lot of mess. So we don't worry too much about alpha. Beta is the scaling exponent. So if you were to plot this as a log-log plot, log Y versus log W, beta would be the slope. And this is how people measure this. So very good question, because yeah, alpha is a bit messier to explain, but beta is not. And one more thing, there is also the issue of time, because biological systems operate on the concept of cycles, metabolic cycles, right? Respiratory cycle, cardiac cycles, for larger organisms, and so on. So everything is cyclical, and including the rate of metabolic enzyme reactions. So these reactions that involve ATP production typically have a cycle time of 1 millisecond on that order. In other words, 1 kilohertz is the frequency for metabolic at the microscopic level. Of course, respiratory, here's a pathologist. What is it? Respiratory rate at 16 or 16 a minute, right? Something like that. Okay, so that's 1 cycle. Heart rate depends, of course, if you're stressed like I am now. I'm not. But it's something like 70, right? 60 is slow, 70 maybe normal, 80 increased. If you go to 120, maybe it's not so good for you when you run. So you see, you have cycles, but at the bottom of this hierarchy of cycles, and of course you have daily cycles, and you have the seasons. So and then of course we have cycles of life, but that there is a scaling of these cycles also with size, and so that's what we call physiological time, which scales with exponent 1/4. All right, so that enough of the introduction. How does it connect to quantum biology? And this is through this observation that Lloyd Demetrius, he was a professor at Harvard and the Max Planck Institute. Nomen omen, Max Planck Institute, I think is very appropriate for this kind of thing, quantum biology. And he kind of looked at all these energy producing structures from chloroplasts to bacteria and their membranes, and to mitochondria in mammals. And he concluded that, I'm going to go faster because it's just too much here, that to explain these empirical observations going back to centuries, you need to compare it to what's happening in quantum solid state physics. We talked about the basically crystal lattices oscillating and quantization occurring because there are waves propagating through the solid. And remember, Einstein made a mistake, assumed that all the atoms are vibrating independently. That's true for gases, but for solids, no. You have to have basically standing waves. And when you assume that, then through some mathematical averaging, you obtain the correct answer for the specific heat of solids as a function of temperature, which was measured. Now here we have scaling rate for metabolic rate as a function of size. And Lloyd Demetrius made this brilliant observation that this is completely analogous. At first, I mean, you shake your head and say, come on, are you serious? But it is. The main difference is that in solids, this is an equilibrium system. You're not pumping energy into it. The system is just vibrating as a result of the temperature, constant temperature. For living systems, this is about energy delivery and maintaining the system at a non-equilibrium state. the mathematics of it is based on the following fact that, and this is the paper that you can look up where the first time the concept of quantum metabolism was introduced about 20 years ago. So he used quantum statistics, and this is called allometric scaling laws. Allometric because it's not unitary, it's not linear, it's nonlinear, power of 3/4 basically. And so he assumed, just like Max Planck assumed, going back to the very first lecture, that Max Planck assumed that in order to explain blackbody radiation, electromagnetic waves coming from all objects at finite temperature, you have to assume that energy is proportional to frequency of radiation. And Lloyd Demetrius assumed that the metabolic energy is proportional to the frequency of metabolic enzyme oscillations. These are oscillating enzymes, right? So glucose comes in, ATP comes out. Actually, in oxidative phosphorylation, one glucose molecule produces 36 ATP molecules, but that's a detail. If you change to glycolysis, it becomes less efficient. It's about only 4, I believe, ATP for one glucose, so it's 9 times less efficient. But that would go into your alpha. The important thing is that the energy scales with the frequency of these oscillating reactions. They keep on going up and down. All right, so the rest is calculating the averages, because the metabolic rate is the average of all the cells. For the body, some cells maybe are operating faster than others, but it's the second assumption is that they are actually synchronizing themselves in a healthy body, and this is actually this is a course on medicine, so we now have a connection to medicine very direct. In a healthy body, you have efficient energy transduction, efficient metabolism. You only eat as much as you need. You don't need more. And you produce a maximum amount of ATP from the nutritional intake that you get. Now, pathologies quite often relate to various things like insulin resistance and metabolic deficiencies, and some of the hallmarks would be either obesity, like in diabetes, or, and I'm not a doctor, please excuse me if I'm making a mistake, but I know a little bit about cancer research because I spent 15 years in oncology, and in oncology is the opposite, cachexia, sudden loss of weight, right? And here is an example where metabolism plays a key role, because for cancer patients, the tumor itself is a sort of foreign body which operates very inefficiently metabolically. It switches to glycolysis from oxidative phosphorylation and requires more intake of nutrient to maintain its weight or grow actually faster. So that's the connection. And I wrote a few papers with Lloyd Demetrius. We met and collaborated, and even one on cancer, the metabolism. And later on, I had a student who wrote some papers on this. How so you can say the following, and I'll just leave you with this: that healthy metabolism can be called quantum metabolism because it's the most efficient, the most synchronized, the optimum basically of metabolic transformations that we can achieve. Pathological is classical because it scales with size. Quantum scales with 3/4 exponent, and this was shown actually in this work. I don't want you to remember all these integrals here, but trust me, they are correct. And we obtained a more general result actually here. This is the most general result, and I'll explain, and then we'll move on to the next topic. Namely, basal metabolic rate is proportional to weight of the living system raised to the power. Remember beta, we had a beta exponent, and there was kind of, well, in some cases 1/2, maybe in some cases 2/3, 3/4, mainly 3/4, and that explains why, because D is this physical dimensionality. If it's a three-dimensional system, like we are, then D is 3 divided by 3 plus 1, 3/4. You get the 3/4 exponent. If it's roughly flat or two-dimensional system, as you may imagine, there's a lot of two-dimensional plants would be, leaves, things like that. So 2D is 2 now. 2 divided by 2 plus 1 is 2/3. Now we have a 2/3 exponent. If it happens to be largely linear, I don't know, kind of think of a snake or worms, then it's D is 1. 1 divided by 1 plus 1 is 1/2. So that's the difference between different exponents because of the geometry of the system. And of course, we don't have a cubic organism or a square organism. So it is an approximation to some degree, but at least explains how dimensionality enters into this synchronization of metabolic enzymes. And it's very similar to the result for solids. You have one-dimensional solids and you get specific heat, will have this kind of dimensionality dependence. In fact, I used to teach solid-state physics, and that was one of the assignment problems. So I knew where to get these integrals. And we applied it to biology. So, okay, so that's basically the take-home message is kind of deep at some level and profound. So for biological systems, temperature by and large doesn't matter because most biological systems maintain their constant temperature, but so that's a thermodynamic parameter in physical systems. And then we have heat capacity or specific heat in physical systems, and then entropy, thermodynamic entropy. In biological systems, everything has a time component because it's time dependent. We have the cycles. So you see parameters for a living system will be cycle time instead of temperature. Temperature sets the standard for physical systems, how fast things turn over in biological system is what's important. And then heat capacity becomes metabolic rate and thermodynamic entropy becomes entropy production rate or heat dissipation. So that's what Lloyd Demetrius contributed to this. And this is a very busy slide. I'm not going to explain to you because we have no time and maybe this is not the place, but I will say one thing. Intriguingly, if you go through all these numbers, remember I gave you this anecdote that Max Planck, when he introduced quantization, he put in a fudge factor, the Planck constant, which kind of worked, and then it turned out to be a fundamental constant of the universe, just a fit to the parameter values in the graph. So the question is there a biological Planck constant? Because we have the quantization condition, just like in physics for metabolic rates. So we did this and we calculated what it might be. And it actually happens to be about 11 orders of magnitude larger, something like this, than the physical Planck constant. And the physical Planck constant, if you remember, and maybe this will be a question on your exam, if you remember the physical Planck constant, it actually determines the uncertainty, right? The Heisenberg uncertainty principle tells you that you cannot measure two complementary properties at the same time to a better accuracy than their product being the Planck constant, roughly. So the Planck constant is important because it tells you how accurate you can be and guess what? This biological Planck constant corresponds to the following. The accuracy of energy production to 1 ATP molecule, you cannot be more accurate than that. And the time to produce 1 ATP molecule being 1 millisecond. And that's the product is the biological Planck constant. I've been working on this paper, the whole world will now know, for 6 years.
Wow.
And Lloyd, my good friend Lloyd Demetrius, is never happy with this. He he wants more explanation. So if we never publish this paper, there'll be a record that
Still on video.
on video. Okay, I'll quickly go through this. Let me just say photosynthesis is this quintessential quantum biology phenomenon, but it's a different kind of beast than quantum metabolism, because quantum metabolism treats the entire organism, living system, human or the elephant, as a synchronized whole, mainly from the metabolic point of view. Photosynthesis is, of course, also a metabolic process for plants, algae, and some bacteria that use light energy to obtain, to funnel it to reaction centers and use for propelling some chemical reactions, mainly glucose synthesis. And this is a very complicated schematic of this FMO complex that is instrumental in the energy transaction, capturing photons, a single quantum of light, and exciting electrons, and then tunneling electrons to the proper reaction centers and utilizing their high energy for synthesis. So I would call this quantum physics in biology, not quantum biology per se, because there's almost no biology here. It's just physics, right? In biochemistry. What happens next is biology, you know, how you make this glucose and how the plants grow and all that is biology. But this is just physics in biology. Happens to be a beautifully structured nano device, solar panel, if you will. And this is the gist, these 7 complexes forming this FMO structure, each of which can be excited by a single photon. But what's beautiful, and this is quantum mechanical in this case, is that they share the excited electrons wave function among, remember delocalization, the cloud? I talked about the quantum cloud. This is where it happens. Not everywhere, but it's shared between the 7 of these organic molecules. So that has been very beautifully demonstrated. That's another view of this. And that's another one. And somebody demonstrated that this is like a very efficient quantum search algorithm for finding the most optimized distribution of the quantum wave function. Okay, a lot of things. As and this is cryptophiles. I think that is it. Some fights. So this is a single-celled algae, which of the coast of Australia, that uses and has the structure, among other systems. Okay, again, a lot of interesting quantum physics went into understanding this phenomenon, and complicated lasers were used to find out, you know, how this quantum excited state exists. And one of this was quantum beating. I just want to go back here. This is the key. These complexes are within, so 7 of them are within about 1.5 nanometers from each other, maybe some closer. I don't think there's anyone which is more distant than that. The reason is that if you have quantum particles, which are separated by more than that distance, there is a problem of tunneling over a longer distance than 1.5. It's called the resonance. It's a resonant energy transfer between these two. So Mother Nature experimented and found arrangements which are within these allowed distances and orientations, and created conditions to support this quantum state. It's not very long-lived, by the way, which is kind of amazing, but it's enough to create this electronic excitation, which then is transferred to the reaction center. This is on the order of 1 nanosecond that this quantum state persists, or from hundreds to thousands of picoseconds anyway. Okay, so why am I bringing microtubules now? Because we find, okay, this is, let's see, oh, this. You see the similarity of this network? No? These are tryptophans, amino acids tryptophans, which form a network of 8 tryptophans in a single dimer of tubulin. And tubulin is a protein that makes microtubules. We talked about microtubules in connection with centrioles. Centrioles are made up of microtubules, many other things are made up of microtubules, flagella, cilia, you know. So very important structures in cells, eukaryotic cells. All eukaryotic cells produce tubulin and have microtubules used for cell division. And we'll come in the last few minutes to their potential utility in neurons in our brains. But here we have this structure which resembles, to me at least, maybe not to you, but resembles very strongly the FMO system in photosynthetic structures, right? Except now we have 8 instead of 7 organic centers, which can be excited by electromagnetic waves. In the case of FMO, the photosynthesis, it was visible light. Tryptophans are not easily, well, they're not really excitable in the visible range. They're excitable in the ultraviolet, around 290 nanometers, much shorter wavelength, higher energy. And with my colleagues at Princeton and Florida, we've been doing experiments on exciting these with laser light and measuring the quantum states in tubulin. And this is something again for the world to hear, because we haven't published this yet. But the results are incredibly encouraging, because we found that, first of all, these tryptophan excitations persist for several nanoseconds, longer even than in photosynthetic systems. So that's quite amazing. Secondly, and maybe since I probably won't have time to complete this entire story, I'll say it now. Secondly, we immersed microtubules in the solution with anesthetic molecules, and anesthetic molecules reduce this lifetime of quantum excitations, which makes us think about the connection to consciousness, because we can turn off consciousness with anesthetics if, so there's a lot of ifs, if microtubules in neurons, as you will see, play a role in consciousness, as they do play a role in centrioles, in the simplest structures, which seem to be single cells, respond to light, move towards it, react. Centrioles built on microtubules, and now we have tubulin and electromagnetic radiation causing quantum states, which persist much longer than in even photosynthetic systems. That's quite amazing. Okay, so this is tryptophan, and there's a lot of math here, which I'm not going to bother you with, but you can actually create reliable models for these tryptophan excitations. You basically create a transition dipole moment in tryptophan when you shine light in the 280 nanometer to 305 nanometers, a range of wavelengths. And that excitation then can interact with other excitations in the neighboring tryptophans, and then you create this collective state. And you can imagine that a collective state could then persist through the entire microtubule. These are some results of these experiments showing you how it kind of gradually decay, so I have to re-excite it. And I'll skip that. Yeah, maybe not. Maybe I'll just say one more thing. So just to finish the thought, what we believe is happening, and I hope you remember I said that not all of biology needs to be quantum. It has to have some quantum states that are important for some function. And in this case, maybe the function is subtle interactions with the environment and communication within the system. And if this could be quantum, could be very efficient, could be incredibly synchronized, and so on and so forth. The problem with tryptophans, slight problem with tryptophans, is that ultraviolet is high energy, and it's hard to imagine, other than maybe by these reactive oxygen species recombination, that living systems would produce on their own quantum excitations or photons in this range. We don't shine ultraviolet on our own, right? Unless you're radioactive. Well, that's even higher energy. So we did another experiment, although that's a glimmer of hope that there is quantum communication. And this experiment involves exposing tubulin and microtubules in solution to visible light now, laser light in the visible range, violet actually, 400 nanometers or so. And so we did. What we found is actually now we have something called delayed luminescence. So we shine light on a sample for 2 minutes. Low intensity, actually. This is low intensity, like naturally occurring amount of light. Then you wait for 10 minutes or maybe 20 minutes, and then you measure with a photodetector re-emitted light. You would expect something to be, well, nothing to be re-emitted, or if re-emitted, it would be right away, right? But no, it goes on for seconds, and that is quite amazing. This is called delayed luminescence. There are some structures which luminesce and phosphoresce, but these are the fundamental units necessary for all eukaryotic cells, which perform so many functions for transport, cell motility, cell division, and we believe also information processing in our brains. This, by the way, is, so I'm jumping fast forward to the last topic. We have about 20 minutes or so, and that's the link to it. How many of you have read or seen the book by Roger Penrose, Shadows of the Mind? No? So Roger Penrose is a Nobel Prize winning physicist. He got a Nobel Prize in 2020, 2 years ago. And more than 25 years ago, he wrote two books, one of which is The Shadows of the Mind. I think it's the first one, in which it's a very big, thick book, very difficult to read, but he basically provided a barrage of arguments that human consciousness must use quantum physics, otherwise we cannot explain too many things, how the brain works. And one of which is it's non-algorithmic. We can find solutions almost instantaneously to some problems which computers would take forever. In fact, sometimes computers cannot solve these problems at all. And he gave examples of some chess games that a good chess player can solve in a second, knows exactly what's going on. A computer is simply trained on algorithms. That this is one example. Some patterns, jigsaw puzzle and puzzles. And Penrose actually is a mathematical physicist who contributed a lot to things like tiles, patterns in tiling patterns in geometry, and also black holes, cosmology, a lot of interesting stuff. And with Stuart Hameroff at the University of Arizona, who's an anesthesiologist, they proposed about 23 years ago or so that not only is the human brain based on quantum computing, but actually microtubules which we talked about, are the computing elements in that structure. And they must be, at least some of their states must be quantum states, and they interact quantum mechanically. So these experiments that I told you about are intended to demonstrate that indeed tubulin and microtubules can be quantum. And so that leads to, all of this leads to some very profound questions. You know, what is really consciousness, and also where is memory stored in the brain? Where is it? Nobody knows really where memory is. And then also could resolve some issues such as amazing computational power of the brain, especially vis-a-vis the energetic requirement of the brain, which is minuscule. And some follow-up questions about, you know, how is this information processing taking place, and what type it is, and so on. So that's the background to it. Kind of for amusement, I'm quoting this guy, Robert Epstein. He posted on this website a statement that the human brain does not do any computing, is not a computer, is empty. The empty brain doesn't retrieve knowledge or store memories. In short, your brain is not a computer, which I think it's nonsense, because I believe that not only the human brain is a computer, but every cell is a computer, and the human brain is a quantum supercomputer. And so we looked at some of the issues, including this incredible computational power. So let's now talk about the numbers a little bit. We have on average 100 billion neurons. So neurons being the computing cells. Until now, we think that the operation of the brain is based on action potential, right? Ionic waves being spikes, basically, electrical signals propagating through axons and triggering others. So interactions with neurons, there is on average 10,000 synapses per neuron. So each neuron talks to about 10,000, sometimes 100,000. Some neurons interact with 100,000. So that would mean that the brain has on the average 10 to the 15th synapses. That's an enormous number of connections. And that gives an incredible computational power, even if it's classical, let alone quantum. There are actually some papers by very well-known neuroscientists claiming that the neurotransmitter release at synaptic connections is a quantum process. So ions can be in two states at the same time, could be a wave function. John Eccles, I think he got Nobel Prize, wrote a paper about this. So by comparison, so you can have the same number of operations. Now, let me backtrack. If you assume that each synapse can operate on the order of 100 milliseconds or 0.1 second, then you get 10 to the 16th operations per second in the brain. But that's conservative, because you could have much faster operations between synapses, maybe 10 milliseconds or even less. But even conservatively, if you take 10 to the 16th, this is 10 times more than the Blue Gene, the supercomputer that IBM created to beat Kasparov at chess does. Okay, but what's not okay, just okay, is that the fact that this Blue Gene computer uses 1.5 megawatts of power, which is about the same as the city of Red Deer. And our brain uses about 25 watts, which is less than any of those bulbs. And for cognition, for cognitive functions, it uses about 5 watts, which is like a flashlight. That's the power that is needed. One battery or two for the flashlight is sufficient to power all your cognitive abilities, including Albert Einstein's. So, okay, so much for comparing apples and oranges, right? I mean, yes, maybe the same number of operations, probably even more, but at what cost? And we, this is the state of our technology. If we can create a supercomputer that beats one human being at chess, and that human being is eating an apple and thinking about his wife and doing some other things on the way. So we should be quite proud of our brains. That's what I'm trying to say. So the questions emerge, is this a quantum computer? I kind of indicated to you that probably is, at least to some degree. It could be a combination of classical and switching to quantum. For example, some of the autopilot activities, automatic responses may not be quantum, maybe simply learned responses. Some may be more, I would say, more creative thinking would correspond to quantum search algorithms, because we can find, some of us may find incredible ideas, almost out of nowhere, not step-by-step laborious search for a solution. How do I, improve my driving or golf game or something like that, many trials and errors, but some people come up with a genius idea, right? So in this case, I would say quantum computing, even some autistic people, people with autistic syndrome, they can do some incredible mathematical operations very fast, right? Almost instantaneously. So things of this nature could be, of the creative geniuses, Van Gogh's paintings and so on. How can you say that this is, slow and painstaking effort? I think it's an almost instantaneous solution to something. Okay, so that would, in my opinion, fit the bill. Some people propose that our thoughts are actually wave functions, and the microscopic wave functions in the neuronal system in the brain, and you may have many thoughts at the same time. I'm quite sure that you do. Even when you just listen to me, you may be thinking about something else, maybe several things at the same time, right? No problem. And then when the thought crystallizes, let's say you have to say something. It's like a quantum wave function collapse. That means from this fuzzy thing, it becomes concrete. The answer to a measurement problem. And I found, I don't know about you, but I found one thing, that when I think about something, I sometimes have a feeling that I know it, but when I say it's something else. When you say it, it's different than what you thought about, right? Did you have that impression sometimes? Yeah, when you have to say it, it's almost like this quantum measurement problem. It's the collapse of the wave function. Before you say it, it's roughly this, but then when you have to say it, it's precisely that. So this may have some implications for AI, artificial intelligence, because of course you can think of future extensions of AI to quantum computing, perhaps, but at the moment I think this would be very difficult to beat if the brain is based on quantum computing. Then the current state of the art in quantum computing, I think, is something like 17 qubits. A qubit is, the quantum equivalent of the bit of information. It's a quantum bit of information. And maybe I'll just say one thing. Classical bits are 0 and 1, right? Up and down. Two possibilities for each bit. Quantum has a phase, not just it's like the complex number. So it's an angle, 2 pi, so 360 degrees. And suddenly you have the computational power, which explodes if you go into qubits. So 16 or 17 qubits is as powerful as probably, I don't know, gigabytes of classical computing, maybe more. Okay, so this is the schematic of this Penrose-Hameroff model, a quantum model of consciousness, which is called orchestrated objective reduction. So it's a mouthful. What does it mean? It means, according to them, that the computing or conscious units in the brain are neurons, and in particular microtubules in dendrites, not so much in axon, but dendrites, because it's known that dendrites are more active in learning and cognition and information processing, things like that. And orchestrate, that means that there is a collective quantum state, just like in solid, I can't keep coming back to solid state physics where you have the crystal. It's not just one atom, but it's a whole orchestra. The whole orchestra is playing. So all these tubulin dimers are in an orchestrated quantum state. And objective reduction means that the conscious moment occurs spontaneously. You know, we may have conscious, well, when we think, right now it's not spontaneous because I think I'm feeding you some information, you're digesting this, but you don't have to, you don't have to have anybody around you to have conscious moments, right? Just think about it, close your eyes and thoughts come in. So this can be, that's objective, not subjective, objective and spontaneous. So reduction, and here is the clincher, which I think is a little bit difficult to swallow. The explanation for this objective reduction given by Penrose, who is a cosmologist, is that these quantum states of tubulin collapse to a classical state as a result of gravitational interactions. That is, and it's very difficult to explain, but I'll tell you. Tubulin can have a slight shift in the center of gravity. It may exist in a superposition of two states, let's say slightly up and slightly down, and these two states interact with each other, and then after a characteristic time, collapse on the real state, which is where tubulin is. It's very difficult, I grant you that, and it's not proven, so you can take it with a grain of salt. But if you read the papers, that's how they explain it. And they found that you have to have so the question arises, is a tubulin dimer in a flask, in a dish, thinking about something, right? Or any of the protein? Well, not, because if you have a single dimer, this collapse time is so fast, like 10 to the minus 30th seconds, that there is simply, just like the quantum wave function of a large object, let's say a car, the quantum wave function is so tiny that you don't see the difference between the separation between two wave functions. So you have to have a large enough quantum state to be long-lived, sufficiently long for things like thoughts to occur. Let's say millisecond time scale, at least. And they kind of did the calculation and they came up with a number which is in correspondence with the number of microtubules in the brain and neurons and all that. I'm not going to go there because honestly, I have some problems with that, but you'll find it in the literature. It is the only fully developed fundamental theory of consciousness, so I grant them that. But there are still some wrinkles to be ironed out, and to me the difficulty in all of this is integration at different levels, from tubulin to microtubules, from microtubules to bundles of microtubules, as you can see here in neurons, and from bundles of microtubules to the entire neuron, and then how the neurons interact with other neurons. So you have so many layers of this consciousness cake that any connection between two layers takes years and years and painstaking experiments to find. And that's why we started with tubulin and tryptophans and quantum states in tubulin, shining laser light, and trying to find these states. How long do they live? And then maybe by the end of the century we'll come to the top. No, I'm kidding. The idea is we do it faster than that. You just have to find the connections. So maybe I'll finish with this. A lot of numbers here, but I think they are very meaningful. So for the time being, let's just keep the Penrose hammer of theory in the back of our minds, and just keep microtubules, because they form very regular, very organized structures in all neurons. It seems like they look like units of some kind of computer, and I don't even have to go at this moment to quantum aspects, which there may be, but even the classical aspects are mind-boggling in terms of how much you can encode, and that brings us to memory. If microtubules and neurons were coding devices, you could encode the entire life story from birth to the end of it. Why is that? Because each tubulin dimer can have, and we know that this is from experiments. It's not quantum gravity, imagine. It is serious stuff. It has 4 states of C termini. C termini are these, proteins have C termini, N termini. C termini of microtubules stick out, and they can be each in up or down state. So that's already looks like a bit, 0, 1. So 4 of these states per dimer. Then there may be 2 electronic states. You can excite 1 electron to be either in alpha tubulin or beta tubulin. Then we know also that tubulin dimers exist in at least 2 conformational states. It's a dimer, so it can be like this, bent a little bit or straight. So that could be a mechanical encoding. This could be electronic encoding. This could be electrostatic encoding. And then we can also have phosphorylation states, which is standard biological encoding, right? Phosphorylation states, et cetera, et cetera. When you add it all up, conservatively, you could have 32 states per dimer, and there is 100,000 dimers in a neuron. So it ends up being 1 gigabyte of memory per neuron if you encode tubulin microtubules. Now, of course, our memory sticks are terabytes. It used to be gigabytes. So it was easy to imagine the old-fashioned memory stick. That's your neuron, right? You can encode a lot of papers, right, in 1 neuron, or a video or something like this. Now imagine that you have 100 billion memory sticks. How much can you encode in that? So, in fact, a lot. You can encode 10 to the 20th bits per brain. It's a humongous number, not immense number. It's huge. And by the way, this is a number that John von Neumann 70 years ago or so predicted the human brain can store in terms of memory. So now we at least have a base, kind of molecular basis where this can all go. And you might ask, where does it go when we eventually stop being alive? Well, it could be actually, if it's stored in electrostatic type of interactions, it could actually be emitted as electromagnetic wave with just like your cell phone sending something, message to somebody somewhere. Anyway, I'll stop at this point. It is tantalizing and quite impressive. Okay, and I will stop in 2 minutes just with something more specific, because we wrote a paper which is based on experiments done with mice. Mice were trained to solve some mazes, and then they were given an inhibitor of calmodulin kinase 2, and they forgot how to solve these mazes. So, sorry, I have no time to go through all this. Yeah, this is that. And calmodulin kinase 2 is an enzyme. It's an enzyme that phosphorylates tubulin, actually, in microtubules in the brain. So there is a connection right away, and this is how it does it. It's activated by calcium waves. Calcium waves go with neurotransmitters, and signals then are sent into the postsynaptic connection, and the hollow enzyme is formed, and then it is a bit like a transformer. It walks on microtubules and has 6 legs. It's hexatic, and phosphorylates serines on tubulin C termini. And here, I mentioned C termini before. These are C termini. This is a tubulin dimer. And this is a microtubule. And you have these sticks, little sticks. These are C-termini of all these tubulins. And now you can phosphorylate these sticks, these C-termini, by the enzyme working on it. And we created the computer model. This is the enzyme, calmodulin kinase 2. Very important player in all of this, including Alzheimer's and so on. And so this is electrostatic map of that. And what's amazing is that it fits beautifully into this surface of a microtubule. These are tubulin dimers on the surface, and you can see here how it fits into such a structure. So it really walks on it perfectly in agreement with the periodicity of the lattice. Oh, here it is. This is a cross-section of microtubule with an enzyme working on it. This is a different view along the axis of a microtubule. This is perpendicular to the axis. And as it phosphorylates, it encodes information. So if that was that's another possibility of encoding information. If that was the mode of information storage in the brain, you also end up with 1 gigabyte per neuron, just by, in addition to the other gigabyte that we kind of fantasized about. This is more reality oriented, and that could be a real memory code. So we create these different states, and you can imagine thousands upon thousands of these states on the microtubule, which is very long. It has maybe 10,000 dimers like that, and or more, maybe even 100,000 sometimes. And then you have many microtubules, bundles of microtubules, each of which. So it's almost like an old-fashioned magnetic tape, and you could encode bits of information. So this is the paper we wrote now a decade ago, which turned out to be quite popular. We got coverage in various popular newspapers, including the Globe and Mail talked about it, and Indian Times and Corriere della Sera in Italy. Okay, so that kind of is where we are at. 10 years later, we haven't made too much progress with this, but we made progress with quantum states of tubulin, which what I told you about today is not published, but we are just finishing a paper on this. And we got funding from the Templeton World Charity Foundation, which is supporting consciousness research. So yeah, my time is up.
So, there is all this recent interest in ineffable content, like things that can't be put in words. And thoughts are a little bit like that. So as I understand it, what you're saying is maybe thoughts are quantum, but when you then say something about it, you're
Bingo.
back to classical
Yes,
physics, right?
exactly, bingo. I think that's perfectly
Yeah,
in sync with this. It makes sense a lot.
yeah. So the times when we're dealing with things that can't be put in words yet is much higher order.
Right, words are classical, thoughts are quantum.
Yeah, yeah, very cool, very, very cool.
So if that's the case, then there's again, this could give us another dimension for future technology, right? If we could somehow emulate this in the quantum versions of AI or some other algorithms, sky is the limit, basically.
Yeah, well, AI is getting better and better at identifying ineffable content, generating it itself, and so on. So it's getting there.
Fantastic.
Okay, well, great. It was a wonderful series of 4 lectures, and we look forward to it. So, we're doing special version of the course for Nepal this summer. So
Wow.
I'll see if you want to take part in that. And then, of course, we'll have the regular.
Mount Everest, this comes, with the territory.
Yes, yes. Well, Nepal's a pretty interesting place. Okay.
Thank you. I really appreciate your participation.
Yeah, thank you.